Okay, so a bit when I was putting all the assignments together, as some of you guys may have noticed, so I put like an assignment that was supposedly going to be assigned on Thursday and then due today, and I had to change that. I don't have assignments due like the day after or assigned the day after an exam because sometimes there's not a lot to put on them. So we'll change them up a little bit and have them one assigned today and due next week, and the other one assigned early next week so you can get to it before the Thanksgiving break, and then it will be due on Tuesday, December 4th, I think, which is the last day of class. Okay, so here's where we left off last time, and I said there's like the really cool mechanism by which the DNA is separated, and so when you see the pictures earlier on in this chapter about the transcription bubble, the transcription bubble really forms around the RNA polymerase itself. So remember, if this is the RNA polymerase, this is sort of a cutaway on the inside, and this gray strand, which is actually going to be the template strand, actually goes inside the polymerase where the catalytic core is located. There are places for the DNA to come in. There's a little hole in the side for nucleotides to come in, and there are exit channels not shown so well here, but one exit channel for the RNA to go out of, because remember, you're only going to have about 9 to 12 bases. I think it's like 12 of the RNA actually base paired to the template DNA, and then the template DNA goes out around the back. Everything that's shown here with a dotted line is outside, so except this. I mean, you're coming in the front here, and then the non-template strand, this red strand, is going to go around the outside of the polymerase. So again, if the polymerase is like this, the template DNA is coming in here, the non-template strand goes around the outside, and the template strand goes through the middle and then comes back out and meets the non-template strand out on the other side there. And so the transcription bubble is sort of like has the polymerase completely surrounding it. So the DNA is only single-stranded as it's going through or around the polymerase, depending on which strand you're talking about here. All right, so last time we talked about the two methods, row-dependent and row-independent for transcription termination. Here we'll talk about the mechanism that eukaryotes do, and I like this because it's the retro Pac-Man mechanism. I didn't have a lot of quarters back then, but the ones I did went into the Pac-Man machine. Well, that and Galaga, which is the best game ever. Just saying. Maybe not. I can't play the modern video games where you're like a guy running and shooting and stuff. It's like I try it on a friend's game. He's like, "Okay, you're dead. Let's try again." "Okay, you're dead." I'm like, I can't figure out how to work the controllers. Anyway, so a simpler game for a simpler time. Anyway, so the process by which the pre-messenger RNA is processed, which we're going to talk about right after I'm done with these couple of slides, is linked to the mechanism of transcription termination. So unlike the mechanism in prokaryotes, in which the last little bit of the RNA is yanked out of the polymerase by either the action of Rho, which is going up there and using a motor activity to pull it out, or the intrinsic row-independent termination, where a hairpin forms and the action of the hairpin pulling against the RNA polymerase yanks the RNA out of there as well. So in this case, you're coming down, here's the pre-messenger RNA, because as we're going to see in just a second, and I sort of hinted at, in eukaryotes, the messenger RNA has to have 3 processing steps done on it before it can be released from the nucleus. There is a site that has the approximate sequence AAU, AAA. This is called the cleavage site. And so when you pass that, special proteins that are actually riding on with the RNA polymerase jump off, go to there, and shortly after, I think like 10 or 12 bases after the AAU, AAA, then you will get cleavage and the RNA will actually get cut. So cleavage exposes a 3' end here that we're going to see is where the enzyme poly-A polymerase is going to add about 200 or so A nucleotides, poly-A tail, which you may have heard about before. And that also exposes a 5' end on this little piece. So this isn't a small piece right here. This is not the scale. There would be the full RNA all the way out, you know. So this exposes a 5' end. And a protein called RAT1 gets on there. RAT1 is a little bit different than the Rho protein in prokaryotes. While Rho is like RNA, or it's like DNA helicase, where it wraps around the RNA and moves up and then pulls the RNA out of there. RAT1 moves up by chewing up the RNA. So it's actually an exonuclease, a 5' to 3' exonuclease. And RAT1 activity is faster than polymerization activity, so very soon the RAT1 protein catches up to the DNA polymerase, and then the complex of polymerase, DNA, and RNA is broken. And once the RNA is removed from the DNA, just like in prokaryotes, the DNA or the RNA polymerase will fall off, and the transcription bubble here will collapse, and that's it. Yeah. So this little bit is going to be like just a few nucleotides. The assumption I would make, my best guess would be that as RAT1 grabs a hold of it and it comes out, it finishes chewing it up into nothing. But even if that were not to happen, this would be released in the nucleus. So as we'll see in the next chapter, there's quality control for eukaryotes in that translation only happens in the cytoplasm. So this little piece of RNA would not have the 5' cap or the 3' poly-A tail that mature messenger RNAs do. So it would never get exported from the nucleus, and it would eventually be recognized as not a legit messenger RNA, and it would be degraded by some other nuclease in the nucleus down to ribonucleotide triphosphates. So one of those two. I should have left the course open here. So these are on the website, but I'll show them here, but you should go back and look at them again as you're studying. It helps once you read everything over and look at the 2D pictures to look at these three-dimensional pictures as well.
The central dogma of molecular biology, DNA makes RNA, makes protein. Here the process begins.
Sure if
Transcription factors assemble at a specific promoter region along the DNA. The length of DNA following the promoter is a gene, and it contains the recipe for a protein. A mediator protein complex arrives carrying the enzyme RNA polymerase. It maneuvers the RNA polymerase into place. inserting it with the help of other factors between the strands of the DNA double helix.
Narrate what's going to go on then. I'm
The
not
assembled
sure
collection of all these factors, the central dogma of molecular biology, DNA makes RNA, makes protein. Here the process begins. Transcription factors assemble at a specific promoter region along the DNA. The length of DNA following the promoter is a gene, and it contains the recipe for a protein. A mediator protein complex arrives carrying the enzyme RNA polymerase. It maneuvers the RNA polymerase into place, inserting it with the help of other factors between the strands of the DNA double helix. The assembled collection of all these factors is referred to as the transcription initiation complex, and now it is ready to be activated. The initiation complex requires contact with activator proteins, which bind to specific sequences of DNA known as enhancer regions. These regions may be thousands of base pairs distant from the start of the gene. Contact between the activator proteins and the initiation complex releases the copying mechanism. The RNA polymerase unzips a small portion of the DNA helix, exposing the bases on each strand. Only one of the strands is copied. It acts as a template for the synthesis of an RNA molecule, which is assembled one subunit at a time by matching the DNA letter code on the template strand. The subunits can be seen here entering the enzyme through its intake hole, and they are joined together to form the long messenger RNA chain snaking out of the top.
If it's eukaryotic, not shown in this video, but there would be introns in there, and so the introns would be being spliced out as transcription is taking place. So it's not always in the exact order in which the exons and introns come flying out of the polymerase there, but it's pretty close. And I wish the video would show it, but imagine that when you see this. So there's release of the polymerase from the initiation complex. All these proteins would just fall apart, and then they would go to bring another polymerase to the transcription start site. Now when you see this whizzing on down the DNA making RNA, try to think that this is only one that they're showing you for purposes of simplicity. This would be like a highway of polymerases racing down, one right after the other, probably as close as they can be to each other, and still have everything working. So you'll be having multiple polymerases making multiple RNAs at the same time. Think back to that figure from earlier in the chapter where you see like one DNA and then you see many RNAs, molecules coming out from either side.
Contact between the activator proteins and the initiation complex releases the copying mechanism.
So you think how close those
the RNA polymerase unzips a small portion of the DNA helix, exposing the bases on each strand. Only one of the strands is copied. It acts as a template for the synthesis of an RNA molecule, which is assembled one subunit at a time by matching the DNA letter code on the template strand. The subunits can be seen here entering the enzyme through its intake hole, and they are joined together to form the long messenger RNA chain snaking out of the top.
I'm sorry about the sound in here. I'll have to ask if they can look at it. I should get out, like play the organ like in an old theater. And the polymerase is leaving the promoter. All right. All right. So now we move on to RNA processing. So doing the 3 processing steps that I'm going to talk about is a strictly eukaryotic phenomenon. We'll come back to see this later on, but I just thought it'd be cool to show this picture right at the outset. So this is an RNA molecule here, the yellow parts, and from here on around there used to be the green part, used to be between here and here. This is an intron that does not code for protein, and it is being spliced out, and one exon over here and another exon over here that do code for protein are going to be joined. This is going to be joined from there to there. So this green part was all hooked up, and now it's going to be cut out. And you can see right there, that's going to be something important. Remember, I keep harping on RNA has that 2' hydroxyl group. There's a special nucleotide in there called the branch point A. It's an adenosine nucleotide, but this nucleotide at this point in this intermediate stage in splicing actually has, so going this way, it will have the 5' and 3' phosphodiester linkages, but when you look around right there, this branch is actually hooked and it has a 3rd phosphodiester linkage that's directly to that 2' hydroxyl group. And so if you did not have that in RNA, then splicing would not be able to take place. All right, so here's the slide I always show. We've talked about DNA replication. We've talked about making an RNA copy of DNA, so the process of transcription. Now we're going to talk about RNA processing. So in eukaryotes, making what's called the pre-messenger RNA that matches the sequence that's transcribed directly off the DNA into RNA that can be sent out into the cytoplasm out of the nuclear envelope and translated by ribosomes into amino acid sequence of protein. So originally there was this concept called collinearity, and that makes sense. So they thought that genes and proteins were collinear. That doesn't mean that the exact number of bases in a gene would match the number of bases in a protein, but the number of amino acids in a protein, if I said bases, the amino acids should be directly proportional. So scientists at the time did not know how many bases in nucleic acid would code for an amino acid, but there should be a standard set number, and then you could divide the messenger RNA by that number and figure out how many amino acids were in a protein. So it turns out through much work that's really cool work, but we don't have time to get into it in this class, that the number of bases in a codon or a word that specifies for each amino acid is 3, and we'll talk about that in the next chapter after this. But each 3-letter word specifies one amino acid that's going to be added to a protein. So why was this wrong? Even in prokaryotes and bacteria and archaea, it's wrong. And some archaea are going to have introns, as I'll mention it now in case I forget to later. But both prokaryotes and eukaryotes have 5' and 3' untranslated. We've already talked about that when I talked about transcription in prokaryotes. The 5' untranslated region is all the nucleotides in the messenger RNA before the 3-letter A, U, G start codon. So every messenger RNA has a bunch of bases in it at the 5' end that don't code for anything. Sometimes they actually don't do anything. Other times they can fold up, especially in prokaryotes, into elaborate 3-dimensional structures to make switches that will allow the messenger RNA to be translated under some circumstances and not translated under other circumstances. And there's 3' untranslated as well. So 3' untranslated is any sequence in the messenger RNA after one of the 3 stop codons, UAG, UAA, or UGA, right? And so there's going to be some sequence at the 3' end. But in prokaryotes, in bacteria, everything in between the start codon and the stop codon is these 3-letter codons, and the coding sequence is not interrupted, so to say. In eukaryotes, introns also occur. So these are sequences within the gene or within the coding region itself, after the start codon, before the stop codon, that don't code for protein. How can you tell that this is the case? So we've talked about DNA hybridization before, so you can take DNA and heat it up to a high temperature. That melts the DNA. Melting means we separate the 2 DNA strands, and if you let the DNA cool back down slowly, the base pairing will reform naturally and the DNA will come back together. That's just if you have the DNA present only. You can also do what's called hybridization. So a hybrid is a combination of 2 things. In this case, we're taking a eukaryotic organism's DNA, which can be purified separately from its messenger RNA. So we've talked about pre-messenger RNA that needs to be modified. That's in the nucleus. This is messenger RNA that's in the cytoplasm, already have all the introns removed from it. If we heat these up, the DNA strands will separate. RNA doesn't really have to, but any secondary structure there will separate. Then let them cool slowly together. Sometimes the DNA strands will fight each other and come back together, and sometimes the DNA will find RNA. And in this case, you can see the differences between the DNA and the messenger RNA. So messenger RNA here is after introns have been removed. And the DNA will bind here and there, and you can see whoosh, like that, right? A little horseshoe shape. That little strand of DNA there is separate. That's a single strand of DNA. It's not bound to the messenger RNA because that part of the DNA is an intron, and that sequence has been spliced out or removed from the messenger RNA. So this is the book picture. Now this is from an earlier edition of the textbook because I like the way this looks better than the one that's in I think the 5th and the 6th edition. So this is an electron micrograph, and this business in here in the middle is DNA hybridizing with messenger RNA. It's a little confusing, but if you look where the pointer's going, see this way here, that's the DNA / RNA hybrid double-stranded molecule. Looks like that. So exons within the DNA hybridize with the messenger RNA, but then you see all those loops on the side that I've gone and highlighted in red here. These are the intron sequences that are present in the DNA but not present in the messenger RNA because they've been spliced out. If you notice a little bit of a difference between this and what was shown on the previous slide is that the size of the introns vastly exceeds the size of the exons. So look, from right here to there, right, just this little bit, just that little bit of real messenger RNA there, if it was a pre-messenger RNA, it would have this loop, then that loop, then this big loop, then that other loop. Each of the loops, even this little tiny one here, is way, way bigger in size than the messenger RNA itself. It's why our genome, or the genome of eukaryotes, is so much bigger than that of prokaryotes. And there's more space between the genes, but there's also space inside the genes of these introns that are not actually coding for proteins. And it's why genomes increase in size as you go up the evolutionary tree, even though the number of genes don't increase significantly. So C. elegans has about 19,000 genes or so, maybe a little bit more than that. The genome is 102 million with an M bases. Humans have 20-some thousand genes, less than 30 for sure, I think less than 25. In the Human Genome Project, when it was started back in the 1990s, I was in grad school at the time, and they kept having, as more of the Genome Project was finished, estimates for the number of genes in the human genome. And the starting number was 100,000. And every time they got more and more and more of the genome sequenced, they're like, hey, there's no genes in here. It's like 100,000, 50,000, 40, 30. Now I think it's in the like 20,000s. I would think it's between like 20 and 30,000, but I'm not exactly sure. So less than 10,000 more genes than C. elegans has. But whereas the C. elegans genome is 102 million bases, the human genome is 3.02 billion bases. So that's like 30 times, 30-fold more DNA for not that many more genes, right? This is a big part of the reason right here. You have a question? Do introns do anything?
Is there a function behind them, or are they just taking up space?
Well, we'll find out later. So we'll find out later in this chapter that one of the things introns allow eukaryotes to do that prokaryotes can't do is to make more than one, and in sometimes tons of different proteins out of the same gene. How are we so much more complicated than the C. elegans worms with not that many more genes? It's due to being able to make this many proteins. And so we'll see that we'll see coming up later that when you take the introns out and stick all the exons together, choices can be made. Sometimes different exons are used in one area and not in another area. And also, and this is important evolutionarily. So remember I talked about, well, I don't think I mentioned this, but when I was talking about transcription factors, right? They have a domain that will bind to the DNA and recognize the sequence. Well, the same domains for DNA binding are used by a lot, sometimes hundreds of different transcription factors, each one of which has only a little bit of difference in this different sequence it can bind. So there's two ways in which this can happen. The first is the whole gene can get duplicated and a copy made. We talked about duplication earlier. And then the second gene is allowed to evolve or diverge from the sequence of the first one without compromising the function of the first one. The second thing is that in a lot of cases, each domain of a protein, and domains in a protein are domains that do a specific job. Like transcription factors will have a DNA binding domain, which is separate from the domain, say, that does transcriptional activation or transcriptional repression, turning it off. Domains are oftentimes packaged neatly into either one or two or three exons. And so you can take the domain from one gene and copy it and paste it into another gene and have like the DNA binding, the activation ability of this transcription factor copied and pasted over here into this other transcription factor gene. Now both of them are activators, but one of them recognizes one DNA sequence, and this second one recognizes a different DNA sequence, right? So this phenomenon, which we don't talk about here, is called it's called exon shuffling, and that's a big reason. So if you had the protein coding all stuck together, straight on, like in prokaryotes, it'd be really hard to take a piece here and put it and copy it and paste it over there, because you would mess up the reading frame, which we'll talk about later. These words come in 3 letters, and there aren't any spaces between the letters. If you take a piece of this and stick it somewhere else, and you don't get it exactly in a set of 3, you screw it up and it doesn't work. Also, you screw up the protein it came from too. So, as we'll see, there's a lot of effort that goes into this splicing process. So the spliceosome, the protein and RNA complex that that carries out this function, is second, I would say, only to the ribosome in size and complexity. A lot of energy is taken with splicing, so if there was not tremendous evolutionary advantage in having it, it would be gone. So here's an example of just straight up what I would call regular splicing. And you can see here that the exons numbered in black across the top and darker colored on the figure are much, much, much smaller than the introns. When you transcribe out of the DNA, the RNA will match the sense strand up here in the DNA and be complementary to the antisense or template strand down here at the bottom. This is the pre-messenger RNA. Then in the nucleus, you put on a cap that we'll see on the 5' end, a poly A tail on the 3' end, and also you remove all of these introns, the sequences that are lighter in color and shown in blue. And it's a lot smaller. Now this is normal. Now 6, for whatever reason, 6 and 7 are missing there, but you see the lines, and I just noticed that they're gone. I'm going to have to look and see if, I don't think I have the figure messed up, but we'll see. But you're using all of the exons. Before the end of this chapter, we'll see that in many cases, all of the exons are not in fact used. Some of them can be skipped. And then in different tissues, some of the exons can be used, and in other tissues, other exons can be used, and that's not possible when the whole thing is stuck together with the introns not there. Yes? Exons are the part that codes for protein. The exons are the parts that have the potential to code for proteins. Always. It's just like, so let's say, we're going to talk about alternative splicing later, but since you're bringing it up, let me talk about it now. All right, in tissue number 1, this happens, and you get exons 1, 2, 3, 4, 5, 6, 7, and 8 all put together to make this messenger RNA that now the ribosome can translate all the way through that, and everything in there codes for protein. But in another tissue, exon 4 is not needed. So instead of splicing from here to here and taking intron 3 out, and here to here and taking intron 4 out, you would splice from here to here and take intron 3, exon 4, and intron 4 all out as if it were one big intron. Even though that happens, exon 4 still counts as an exon because it has the potential to code for protein. You would just say that that exon is not being used in that tissue due to alternative splicing. Does that help? And I'll talk about it more when we talk about alternative splicing later, so come on back and ask another question if you've still got it at that point. All right, so as long as we're talking about what is, right, what is a gene? I've sort of touched on this before. So a gene is all of the parts in DNA that gets transcribed into RNA, whether it is pre-messenger RNA that will become messenger RNA, or in prokaryotes just messenger RNA straight up. This is the only RNA in this whole list that gets translated into protein. But all these other RNAs, even though they don't get translated, have an important job to do, and the cell would be in trouble and the organism would be in trouble if they did not if the organism did not transcribe them. So in addition to the part of the DNA that just gets transcribed into RNA, any sequences that are necessary for that transcription to take place. So promoters, enhancers way far out, even like 100,000 base pairs away. So you wouldn't say that whole thing is part of the gene, but you could say, hey, this enhancer is part of it way out there. Sometimes there's sequences, I didn't mention this in the transcription chapter, but just like there are enhancers and silencers which turn transcription on or off respectively, really far out, 5' from the gene, there's also the same thing 3' from the gene too. So you can have a sequence 100,000 base, 75 or 100,000 base pairs, 3' from the coding part of a gene where transcription factors bind and influence whether the gene gets turned on or off. So all of that. So here's a prokaryotic gene. We talked about the 5' and 3' untranslated regions. Everything in green here is RNA sequence that gets transcribed from the DNA, but only the green part between where it says start codon and stop codon actually codes for protein. The 5' untranslated and 3' untranslated sometimes are used for regulatory purposes, sometimes not, and they're just there. Also, only in prokaryotes, there's what's called the Shine-Dalgarno sequence. Again, did they call that in microbiology, or they call it a ribosome binding site? All right. So it looks like they're trying to get it to the modern thing. So ribosome binding site, or RBS, we'll see in prokaryotes there's a specific sequence that is recognized by the ribosome, and that's going to be different than how things work in eukaryotes. And you have that whole thing. No introns are present in here. Like I said, all of this codes for messenger RNA because bacteria don't have introns. Some archaea do have introns, but they're weird enough that we're just sticking to the basics here. We're going to talk about bacteria and eukaryotes, and archaea can be, saved for a more advanced class. All right, so here's my stealing of the same figure and making it into a eukaryotic messenger RNA. So no ribosome binding site. I didn't want to take the green and just cover it up. I should probably take it and try to put it back together, but I'm lazy, so this is what you get. You also have in eukaryotes a 5' and a 3' untranslated region, or UTR. You can just put 5' or 3' UTR, all capital letters, and that's fine. And then you have the start codon and the stop codon. This is the area, the protein coding region, but unlike in prokaryotes, this is going to be interrupted by introns that need to be spliced out. So this would be in eukaryotes pre-mRNA. Pre-mRNA is the sequence that matches the DNA exactly and what gets transcribed initially inside the nucleus. Then inside the nucleus, these 3 processing steps that I've already hinted at a bunch of times are going to take place. We're going to add a 5' 7-methylguanosine cap, which we'll show you a picture of in just a second. And then we're going to add a poly A tail on the 3' end and remove the introns. All 3 of these steps mark a messenger RNA as being mature, and all of these are required by the cell in order to gain the ability to have that messenger RNA exit out of the nucleus. It's a quality control mechanism that keeps messenger RNAs that are improperly processed from getting out into the cytoplasm, and it works pretty well because ribosomes are only in the cytoplasm. Whether you're talking about the ribosomes that are part of the rough endoplasmic reticulum or the free ribosomes that are just floating around in the cytoplasm, no translation occurs in the nucleus. So if you can keep if you can keep incorrectly processed messenger RNAs in the nucleus, they will eventually be degraded into their constituent ribonucleotides, which can then be made into triphosphates and become part of another RNA. Keeps you from making defective proteins. All right, so I've been harping on nucleic acid polymerization only occurs in a 5' to 3' direction relative to the DNA being polymerized. Here's where you're going to have an exception, but it's not really too much of an exception because it's not nucleic acid polymerization. You're just adding one nucleotide that's going to be a cap. Here's the 5' end of an RNA. It's still got 3 phosphates on it because it was the first nucleotide added. So the 2 phosphates were never taken off because a phosphodiester bond was never formed. First thing that happens is one of the phosphate groups gets knocked off, so then the first nucleotide only has a diphosphate instead of a triphosphate. Then the 5' phosphate is going to attack the alpha phosphate of a regular GTP. And this is weird because it's the 5' phosphate here is attacking the alpha phosphate. That's the one closest to the 5' carbon. And then the GTP becomes added to the 5' end. So there's two things that are different about this. The first thing is that unlike the rest of the chain, there's not one phosphate between nucleotides. There's 3. One gets knocked off, but when you add the GTP, 2 get knocked off the GTP, but 1 is still there, so now you've got 3 phosphates between there. Also, this is the only linkage that is a 5' to 5' linkage. The 5' end here attacks the 5' phosphate of the GTP. So 5' here to 5' here. The 3' end of the base is just sort of hanging out in space over there. The 3' hydroxyl, nothing gets done to it. That's what this is here. So they turn the base, they flip the base around like that from the way you're normally used to seeing it. Here's the 5' carbon, 3 phosphates, and another 5' carbon. And then what is the 7-methyl guanosine? So it's a G, guanosine, and then it's just got a methyl group at the 7 position. I'm not going to ask you to draw this. I'm not going to give you 4 guanosines, each with a methyl group on, and ask which one is at position 7, right? You need to know that it's a 7-methyl guanosine. You don't need to know exactly where that is. It's just this modification happens after a normal guanosine triphosphate is added. That modification is a marker that this is a capping guanosine and not a guanosine, a G base that's just somewhere else in the chain. Doing this will tell the cell in the nucleus that the 5' end is good. Once this messenger RNA gets exported to the cytoplasm, it will also continuously tell the cell in the cytoplasm the 5' end is good. Because in some cases, you can have a messenger RNA that gets broken. You know, oxygen-free radicals, a bunch of other metabolites in the cell are capable of busting up phosphodiester bonds, or it just gets sheared apart, or whatever. Something bad happens. And the cell has to be able to recognize that this messenger RNA is now bogus and should be destroyed. And so if it gets broken in half, the 5' end will now no longer have a G on it, or a 7-methyl guanosine, and it gets destroyed. Yes, the 7-methyl guanosine part with a small chain will still be present, but it will not have the poly A tail we're talking about at the 3' end, so it gets destroyed. The cap and the tail tell the cytoplasm surveillance mechanisms this messenger RNA is still good and it's still full length. If either of those goes away, the cell is very, very quick to destroy it because you don't want to make proteins that aren't correct. Good way to kill yourself accidentally. You don't want to do that. All right. So now we're going to talk about the the 3' end, polyadenylation again. And remember we talked about previously that the mechanism of cleavage here for polyadenylation is linked to the way that transcription termination works in eukaryotes. So here's the AAU-AAA sequence. Shortly thereafter, you get cleavage there. There's a U-rich sequence here, which is going to bind to A's in the template DNA. So when, as we'll see soon, or as we already saw, RAT1 moves up and then it can get rid of this because it's all AU bases. But here what we're looking at is you now have a free 3' hydroxyl and an enzyme called poly-A polymerase comes in. Poly-A polymerase is one of the few enzymes in the cell that is capable of polymerizing a nucleotide polymer without a template. How does it accomplish this? It only adds A's. So it will recognize, so not shown here, and over the top for this class, there's proteins that are going to be around here that recruit the poly-A polymerase and say, hey, here's where you start going to town and adding A's. Poly-A polymerase gets on there and just starts adding A's to the end of the chain. Doesn't need a template because all it's doing is adding A's. The tail is about 200 A's long, yes. This is eukaryotes. So eukaryotes get the cap, the tail, and splicing. Prokaryotes, you transcribe it and it's good to go. As we'll see in the translation chapter, in prokaryotes, because there's no nucleus, you can start translating an RNA immediately. As soon as that 5' end of the RNA comes off the RNA polymerase, ribosome can jump right on it and start translating. All right, so how do we know? How does the polymerase know how long to make the tail? I think it's like 200 or so nucleotides, somewhat different for each organism. It's the same homeostasis mechanism that we talked about with telomere length. Do you guys remember how that worked? So telomerase uses its RNA that matches the telomere, or that is complementary to the telomere sequence, and keeps adding more sequence to the 3' end of the telomere until it's long enough. How does it know that it's long enough? Telomere binding proteins will bind to that sequence. Each of them has a very small but real inhibitory effect on the telomerase enzyme. When the telomere gets long enough, the combined inhibitory effect is enough to inhibit the telomerase and make it go away. Same thing here. There are poly-A binding proteins. And each one of them has a very small effect on the poly-A polymerase, not enough to make it go away, but a little inhibitory effect. But when the poly-A tail gets long enough, there are so many binding proteins on the poly-A tail that together their combined inhibitory effect is enough to make it stop, and that's long enough. All right, so I dragged this figure back from the previous chapter that we just talked about there. So here's the pre-messenger RNA. This is way short. It's going to be a lot longer. We're not talking about the splicing that takes place there, but here is the poly-A signal, as we say, and then 10 to 30 bases later, there's the cleavage site. When this gets cut open, you are giving a 3' end with a 3' hydroxyl that is able to be added, have the A's added to it by the poly-A polymerase. But when you cut this, here's the 3' end for polyadenylation. There's also a 5' end connected to the now very short messenger RNA that's still in the polymerase, and that 5' end will allow the RAP1 protein to bind, and it starts using its exonuclease activity until it gets up here, and by the time it gets up there, you've got the sequence there with all the U's, with all the UA base pairs, and that's easy to get the RNA out. So the RNA comes out, and then this ternary complex of RNA plus protein plus DNA collapses. When the RNA comes out, the RNA polymerase falls off, and then the transcription bubble closes back up and snaps shut. Okay, so let's look at the introns now. This is some exon over here and another exon over there. We're just calling them exon 1 and exon 2. It could be exon 5 and exon 6, whatever. The dark green sequence is exon, the light green sequence is intron. Everything shown with the bracket here in intron and in the light green sequence is gotten rid of. At the joint between an exon and an intron, both at the 5' end of the relative to the intron and at the 3' end relative to the intron, you will get consensus sequences. These are somewhat different. Remember, consensus, they could be different. The spliceosome recognizes them. The closer these sequences are to consensus, the better the spliceosome recognizes them and could just start splicing there immediately. The farther they are from there, the more the spliceosome needs help from other proteins, and that's how we're going to do alternative transcription. So the spliceosome is helped to each of these sites by auxiliary proteins that are too much they're described in the chapter, but they're too much for you guys to learn. I want you to learn the process here, what I'm saying in class, not memorize all these proteins that we do talk about in molecular genetics, but it's because molecular genetics is a whole semester of just the second half of this class. The 5' consensus sequence is usually referred to as the splice donor. The 3' is referred to as the splice acceptor. All I want you to know is that generally the first two bases, with some exceptions, but generally the first two bases of each intron are GU, and the last two are AG. Now the GU sequence and the AG sequence, being part of the intron, are going to be gone. So when I have you do problems where I show you like a little piece of DNA and I say, here's the 5' end of the intron, here's the 3' end, what would the spliced DNA look like? You have to take the sequence that starts with GU, the sequence that starts with AG, remove it, and paste the two ends together. So here, let's say this is a C, right? C, A, G, and then it would be G, whatever these are down there. That's what the spliced looks like, because the intron is going to be completely removed. You don't have to memorize these at all. I just wanted to put that in there to show you if you ever have DNA synthesized and you want to look at, you know, here's a C or an A, like there's an A or a G, that's going to be an R, where did it here, R. So instead of just A, C, G, T, when it's going to be like A or G, R, and C or T is Y. These are the two most common. R is for purine, Y is for pyrimidine, right? So if I put an R in a DNA sequence, that means that half the time it's going to be A and half the time it's going to be a G, like a mix. Or Y, CT. If you order DNA, you can get it with variabilities in it. We already talked about this guy down here. N just means any nucleotide. So if there's an N in a sequence, it means you don't know what base should be there, or it means any of the bases could be there. All right, so as I said earlier, the job of removing introns is done by the second largest, I think, molecular machine in the eukaryotic cell, which is the spliceosome. It's composed of many different moving parts, again described in the book, but you're only going to need to know what I'm talking about here in class. The spliceosome is composed of both SNRPs and proteins. Put that in there so I can say SNRPs. We're going to go on a SNRP hunt. Biologists actually spent millions of government dollars going on a SNRP hunt, and they actually found the SNRPs. So SNRPs are small nuclear ribonucleoproteins. So these are a protein plus RNA complex. And there are other proteins that have no RNA complex with them that go together. The spliceosome is composed of many of these different items, and it's just huge. As I've harped on many times, splicing is the actual splicing reaction is catalyzed or facilitated by RNAs, and not by proteins. So as I threw couple new pictures in here later on to show that this whole RNA world that we've been talking about, and the fact that peptide bonds in translation done by ribosomes is catalyzed by RNAs, all this stuff would have been outlandish talk, not that, well, I guess for you guys before you were born, so it is a long time ago. Anyway, but not that long not long ago in the grand scheme of things anyway. Like I said, there are other proteins that help to find the splice sites here. I would also be remiss if at this point I did not also mention this little guy right here, the branch point A. This intron is not drawn to scale. The branch point A is very close to the 3' splice site relative to the size of the entire intron. I don't know, maybe 20, 30 bases away, something like that, versus this could be hundreds or thousands. They just can't put it in the figure. But the branch point A is the nucleotide that's going to make that lasso or lariat looking structure. All right, so let's look at the chemistry of how this works. So the first thing that's going to happen is there's 2 steps in this. It's a 2-step process. The first step is that with the help of the spliceosome machinery, right? This is all facilitated. I'm just not telling you what actually is doing the job. The 2' hydroxyl of the branch point A is going to attack the phosphate of the 5' G. So this is going to be like GU there. So it's attacking the phosphodiester bond that is between the 3' base of the exon in front and the 5' base, the G of the GU dinucleotide at the 5' end of the intron. This leads to this weird structure here where you have the A has a 5' and a 3' bond. So here is the A, there's the sugar, 5', 3', but now there's a 2', the hydroxyl that was formerly here. All the rest of these have an OH there. The OH on the branch point adenosine is bound to the 5' phosphate from the G. This should be a U because it almost always is, but so like GU. These 2 bases are these 2 bases from right here. So this folds over on that to make this lariat structure. Good so far? That frees up a free 3' hydroxyl group there on whatever base is at the end of exon 1. That then goes and attacks the 5' phosphate on the first nucleotide in exon 2. That will join exon 1 and exon 2 together. So whatever base is here, the 3' base here, is joined to the 5' base there, and the 3' base here, that G from the AG, its 3' end is cut off, so it ends up just with a hydroxyl group, and this lariat structure here is released. The exons are spliced together, and this lariat or lasso-like structure is released, and it's chopped up into nucleotides. So ribonucleotide monophosphates that then can be reconverted back to triphosphates to give them energy again, and they can be added to new RNA chains. So this entire intron, the sequence between the two splice sites, is removed, and exon 1 is joined to exon 2. Two-step process. Okay? All right. I want to make sure, so the branch point A is important because if you didn't make this lariat, the first reaction is what actually frees the 3' hydroxyl here to be able to make that second attack. And now I've put back in the picture at the beginning. So this lariat structure right here is this. So this is what you would see after the first attack. So the branch point A there, 5', 3', 2' here, this is the G and the U, GU, right? And then here is the 3' end of that intron, AG. You can see that the spliceosome machinery here has already catalyzed the first reaction, and this is at the point where the free 3' hydroxyl that was freed here is going to attack the 5' phosphate there. And the 3' end of the first exon is going to be joined to the 5' end of the second exon. This yellow joins that yellow, and then the spliceosome components will release this lariat structure, this whole intron here. That's this part, and it's going to get, this is going to all get chopped up, and the spliceosome goes on to do more work, and right here, these two enzymes end up being joined. Good? All right. So, how do we know that the catalysis mechanism of making this first attack that makes this lariat structure and the second attack of the hydroxyl here on the phosphate there are facilitated by RNAs? It was with the discovery in the 1980s of a organism that had self-splicing introns. So look over here. See this green part where it says exon 1, green part where it says exon 2 there? This is part of the RNA sequence. Everything between here, so all this, right? All that light green stuff there is an intron. But you can see all the stems here and loops and other parts of that. This whole thing will actually fold up into a three-dimensional structure that will facilitate the removal of itself and catalyze the splicing of the 3' end of exon 1 to the 5' end of exon 2, all by itself. And there are a couple different mechanisms by which self-splicing works, described in a book. Don't need to know the difference between them, just that it exists. And here's the little warm water pond creature called, the genus is Tetrahymena, and there are a bunch of different species. These were a model genetic organism that was being worked on. And again, models are not being worked on because everybody wants to know about Tetrahymena and the secrets of its life. But by learning how things work with Tetrahymena, you can learn how things work with other creatures, including us. And this organism is, so here's an electron micrograph, falsely colored, showing the outside. You can see all the yellow parts here are cilia. So this is pretty big. I didn't put the dimensions on there, but it said in the material that I looked through that this guy is bigger than some eukaryotic cells, which, you know, for a little pond creature is pretty big. This one is stained with DAPI, which is a stain that that recognizes DNA, so that's the nucleus, and there is a green stain that's against the cilia there. And I'm not sure what the red is for. I would imagine that the red is for one of the cell structural components, but I wasn't able to look, you know, in just a couple minutes it took me to find all these guys. But anyway, Dr. Tom Keck at I think he's still there at University of Colorado was the one who discovered this. And as happens every time you do something that affects or gets onto the central dogma of DNA, he won the Nobel Prize for it. So this little organism got a Nobel Prize. All right. And by showing that this RNA intron could fold up and catalyze its own removal, some of the RNAs that are part of the spliceosome, scientists said, hey, these sequences look somewhat similar. So they don't have the whole sequence that holds it together. The RNAs that catalyze the splicing here are similar to what still exists in eukaryotic cells. It's just that instead of a bunch of RNA holding those RNA sequences in the right spot to do the catalysis, in the modern spliceosome, what happens is that you have proteins that have those RNAs embedded, and the proteins hold the RNAs in the right spot. But without the work on Tetrahymena, scientists would have never known that that was possible. This self-splicing is the first example, I believe, as far as I know, is the first example of a ribozyme activity ever discovered. So ribo for RNA, so ribozyme. So like an RNA acting as an enzyme. And when they found out, hey, if this is in splicing, as we'll see coming up in translation, scientists have known forever that there were a bunch of RNAs in the ribosome that does peptide bond formation in proteins, but they were just assumed to be structural only and not catalytic. But once they found out, hey, RNA can catalyze enzymatic reactions, scientists went back and did crystal structures and looked more closely at the ribosomal RNAs and found, hey, these do it too. So pretty cool. Now, back to what we were talking about earlier, both of you guys. It's like, so what's the point? Why do all this? It's a lot of work. It's a lot of energy. If it's done incorrectly, the mature messenger RNA that gets made is no good. So why are we doing this? Because we want to be able to do RNA or alternative splicing. Here's one example. There's another example in the same figure. Go back and look at that one after class 2. So here's just a simple example. Exon 1, 2, and 3, 2 introns there. This is the pre-messenger RNA. Again, it looks exactly like the DNA. And cleavage of the 3' end, polyadenylation. Not shown here is the 5' capping. In this example over here, introns 1 and 2 are removed, so you get a mature messenger RNA that has exons 1, 2, and 3 all right next to each other. But over here, in tissue number 2, they're going to not want whatever amino acids are contained within exon, or coded for by exon 2. So instead of getting a splicing here, and intron 1 removed, and splicing here, and intron 2 removed, which joins exon 1 to 2, then to 3, you get splicing from the 5' site of this intron. you get the 3' or splice donor here is ignored. The acceptor, I'm sorry, is ignored. The splice donor here at the 5' end of that intron is ignored, and so the splice donor at the 5' of this intron goes to the splice acceptor at the 3' of the next intron, and this whole thing is cut out, including exon 2. And so you get exon 1 and exon 3 by themselves. Is this still an exon? Yes, it is. Why? Because it has the potential to code for something, to code for protein. It's just not used in this case over here. In this case, exons 1 through 3 are all used in part of the mature messenger RNA. They all get translated. Here, only exons 1 and 2 are used. Or 1 and 3, only exons 1 and 3 are used, and then that goes away. All right. Here's another example. So the calcitonin gene is used in both the brain and in the thyroid, but different parts of it are used. In this case, there's an interesting difference in that by removing one of the exons along with the adjoining introns, you make use of an alternative cleavage site for polyadenylation. So here, exons 1 through 6, introns 1, 2, 3, 4, 4, and 5. The pre-messenger RNA in both of these tissues looks the same, but in the brain, instead of getting intron 3 removed and intron 4 removed here, going to exon 1, 2, 3, 4, 5, and 6, like you see here, or I'm sorry, 1, 2, 3, and 5, and 6, like you see here, in thyroid cells, you get exon 1, 2, 3, and then 4 normally, because you get this guy and this guy removed, right? And then as you're getting transcription, before you get to the splice donor for intron 4, you have an AAU AAA cleavage site there, so this gets cut off right there, and you get exon 1, 2, 3, 4 only. Why do you not get exon 5 and 6? Because the messenger RNA is chopped right here, and before these are even transcribed, RAT1 gets on that 5' end, and probably in right about here is where transcription stops. So by an alternative splice, by leaving in exon 4 and not splicing it out, then you get that 3' cleavage site in there that actually makes a shorter protein. All right, so we'll see you in lab today. If you're not in lab today, I'll see you on Thursday. It's a nerve cell versus a hormonal tissue. I'd have to look it up. I don't know.